going to talk about profiling cancer for precision oncology, which has been a recurrent theme. I'm Ben Harmson, and I just want to thank Aki and Hope for Stomach Cancer for the opportunity to speak to you today. This is a topic that I'm very passionate about. I have been a caregiver, and my background's in genetics, so I do have some disclaimers. So let me jump forward here. I am currently employed as an executive director for Covance. Covance is a drug development company and a medical device development company. So we do things like clinical trials for therapies and medical devices to bring them to market. I'm not here representing Covance today. I am representing MyOncopath. I've been on the board of directors for MyOncopath for 3 years now. MyOncopath is a precision oncology telemedicine clinic. So what we do is we support patients and providers with expertise in precision oncology and really the genetic testing that is involved in identifying the markers for targeted therapies. So that today my views are my own views and the views of MyOncopath. Go to the agenda here. Oh. Okay. Sorry about that. Okay, so the agenda today, we're going to talk about the history of precision oncology. I'll highlight some of the trends that are happening today and through the history, and some of the recent kind of accomplishments that have been milestones in this space. I'll talk about biomarker testing. In general, biomarker is a pretty general term. I'll really focus mainly on genetic testing and genomic testing, but we've heard a few of these themes already today, so it'll be kind of reinforcing some of the other things that you've heard. Jumping into the history of precision oncology, the first targeted therapy was approved in 1997, so this field has been around for over 20 years. In 1998, Herceptin was approved for HER2 cancer, breast cancers. In 2010, it was okayed for gastric cancers. So this will be kind of a recurring theme that you will see with targeted therapies. They initially get approved for one type of cancer, but then later that label could get expanded for other types of cancers that also carry that similar biomarker as the original FDA approval for the other cancer type. Keytruda was jumping forward to 2017. Now we're really starting to think about treating patients and cancers based on the markers or the profile of the cancer. So in 2017, we have monumental approval for Keytruda, the first universal approval for all tumor types based on the MSI-MMR biomarker. In 2017, there were actually 2 FDA approvals for large panel genomic profiling, tumor profiling tests. The first commercially available test was approved for Foundation Medicine. They have a Foundation 1 test that has about 18 different FDA-approved treatments that you can determine are available to patients based on that test. 2 of those treatments are immunotherapies. Okay. And then in 2017 and 2018, the FDA approved 2 CAR T therapies for blood cancers. This is important breakthrough for cancer treatment because CAR T therapies are when you take a patient's immune cells out of their body, you can genetically engineer them, put them back in to attack the cancer. These are very effective treatments in these blood cancers. But I was just at a cell and gene therapy conference about a month ago. There is a ton of money, to Dr. Wu's point earlier. The money is coming for more of these therapies. They're in clinical trials for solid tumors now and for gastric cancer. In 2018, CMS, which is Medicare and Medicaid, finalized coverage for NGS testing. This means that the tumor profiling is now covered by Medicare and Medicaid for any stage 3, stage 4 recurring or resistant cancer patients. This is huge because it puts a lot of gives a lot of patients free testing, also especially for gastric cancers, and then also it gives a leadership position for other payers like insurance companies to follow this lead because there's still a slow adoption in some places for covering the cost of this testing. In 2018, the second universal approval for all solid tumors. This one is for a rare marker that's found in about 1% of all tumor types, but it's still a very significant treatment if you have that marker, NRTK marker. In clinical trials, that treatment shrank tumors in 17 different cancer types. And then just jumping to today, there are over 80 targeted therapies approved by the FDA, so there's quite a menu available. It also reinforces the critical need to do the testing. There are over 500 clinical trials in the US for gastric cancers. So there's a lot of combination therapies, as we heard a little bit about earlier, and a lot of targeted therapies and a lot of new markers coming. So it's very good to be aware of what's happening in the latest. We'll jump into the testing here. This is genetic testing. It's hereditary testing. It's important in gastric cancers. About 10-11% of gastric cancers have a hereditary marker in the patients. These are, if you look at the picture here, you have a father who's a carrier of a gene mutation and a mother who's a non-carrier. If they were to have 4 children, in this scenario, 2 of them inherit the father's gene with the mutation, and 2 of the children inherit the mother's healthy gene. We can get into preventative care, which was touched on a little bit earlier, but in preventative care, you can do things like lifestyle changes, which would be diet, exercise, things like that, but then you can also start screening earlier for cancer. And if you can get that earlier detection of cancer, then you have a better opportunity of being cured. Tying it into precision medicine, some of these hereditary markers, like BRCA1, BRCA2, are linked to a group of treatments called PARP inhibitors. PARP inhibitors are FDA approved for other hereditary cancers like breast cancer, ovarian cancer, and most recently it was approved for prostate cancer. It's important just to know what your hereditary profile is as well, for not only the benefit of your family members, but also for treatment decision making as well. There are clinical trials in place today for PARP inhibitors and gastric cancers as well. Okay, jumping forward here to genomic testing. This is tumor profiling. The first is solid tumor profiling. This is the gold standard still. These are larger panels. They're more reproducible. They usually include TMB and MSI, which are good biomarkers to predict if immunotherapy is a good treatment option. You can add the PD-L1 testing as well. So we would generally order a large panel if we had tumor tissue available. And if not, then there's a liquid biopsy, but just back to the tumor profiling, they take the tumor tissue and they extract the DNA. They can sequence the DNA and map it for markers. And if the tumor tissue is not available, then we have liquid biopsy is another viable option. Should I hold for a sec? Keep going. The liquid biopsy is less invasive, which is a nice option if you can't get tumor tissue. It's usually a smaller panel and can be used in lots of different you can utilize it clinically for different tools like monitoring disease progression, response to treatment, and then if there is a recurrence in the cancer, you can pick that up with a liquid biopsy and you could potentially find a new marker to treat with. So just in summary, I think a lot of this was covered today already, but it's critical to assess the type of cancer, the stage of cancer, along with the profile of the cancer. Biomarker testing is helping patients get access to targeted therapies, including the immunotherapies that we've heard quite a bit about. And in some cases, this is improving clinical outcomes versus the standard treatment practices. There are a large number of clinical trials and a very large menu of treatment options that are targeted therapies today that are targeting markers in cancers. So it's important to know what your profile is. And, I think that what we're all hearing today is, talk to your doctor about these types of tests and are they available for you? And if you don't get the answer you're looking for, you can get another opinion, as we heard as well. But this is a very critical opportunity, I think, for patients to get the best treatment and the best care. And then I had provided some, these are just some references and resources that I put together. Some of these are very technical papers to support the talk, but also there's some resources here that you can use to learn more about this discussion. Thanks.